So I want to start by saying that obviously we all know that neurodevelopmental disorders, or NDDs, are heterogeneous, and so are their causes. So heterogeneous, what I mean by that is just they're very mixed. There's no one cause of ASD or ADHD or OCD, and there's no one individual that represents all other individuals with the same disorder. And through extensive research, and also research within POND through the genetics platform, we know that genetics plays a very important role in the development of these NDDs. So we know in the case of autism spectrum disorder, or ASD, that it's highly heritable. Mutations that we find that are associated with ASD are very rare, so each individual type of mutation in specific genes only accounts for less than 1% of all cases. And again, we know that not all cases of individuals with ASD can be accounted for based on a known genetic cause. So only about 25%, or depending on the study, 25 to 40% of cases have a known genetic cause. In the case of attention deficit hyperactivity disorder, or ADHD, we also know that it's highly heritable, but in the research that's been done to date, there have been very few strong and consistent risk genes identified. And then in the case of obsessive compulsive disorder, or OCD, even fewer risk genes have been identified. Yes?
Sorry. I don't know, how can something be highly heritable but yet not have a known genetic cause?
So the heritability comes from the fact that we know that in the family history, there are cases of individuals who have the disorder. However, because we don't fully understand the actual causes of each disorder, we can't necessarily identify a specific gene or a specific genetic component that contributes to that. So that's a huge gap in knowledge, for sure. So in emphasizing that gap in knowledge, we know that genetics is important. We also know that environment is also very important. And this concept of epigenetics is really interesting because it actually is affected by both genes and environment. And so it's a way that we can measure a reflection of the interaction between genes and environment. So this figure, I just want to sort of illustrate a point to demonstrate how epigenetics regulates how genes are read, or how genes are expressed. So we have this sentence, "When I sing well, ladies get sick." That can represent, for example, a gene. And I know that the brain is very important for NDDs, however, other organ systems are important, but for the sake of this example, I'll just use the brain for now. So we see that in these two sentences, I've added punctuation in the green. So we've added 2 commas in different places in the sentence. And those represent epigenetic changes. And these epigenetic changes change the meaning of each sentence, but still each sentence makes sense. So this is within the normal variation. Epigenetic changes are part of normal development. However, when we add punctuation or epigenetic changes in places where it doesn't make sense, you start to lose meaning in this sentence. And this can cause effects that make changes in the brain that can lead to the presentation of, for example, ASD features or ADHD features. And as you continually add additional punctuation or epigenetic changes in the wrong places, we see that these sentences no longer make any sense. And that's how we can lead from improper epigenetic changes to perhaps an ASD diagnosis or an ADHD diagnosis. So the lightning bolts sort of represent the different changes that are a consequence of these improper epigenetic alterations. So they can be in different places, in different magnitudes, and can eventually lead to disease. So what is our goal of the epigenetics platform within POND? So we want to use epigenetics to identify new biomarkers of NDDs, so biomarkers being things that we can measure in patients that can help us to distinguish between individuals with and without an NDD, and also to better understand the underlying disease mechanisms. So in identifying these new biomarkers using epigenetics, We can use these to complement the existing tools that we use to diagnose individuals with NDDs. And we can also use our better understanding of disease mechanisms to improve drug development and also to identify new drug targets. And one of the questions that we're asking is whether or not there are epigenetic profiles that are distinct for each diagnostic category, or if there are some that overlap across disorders. So this figure, I just want to use it as an example to show you how we can use epigenetics to molecularly profile individuals with NDDs. So, for example, in the top group of individuals, you can imagine that this is a group of individuals that have various neurodevelopmental disorders, and we can use genetic or molecular profiling, and in our case, specifically epigenetic profiling, in addition to the usual battery of clinical assessments that are used to diagnose individuals, to try to profile individuals to see which groups of people have more similar profiles to each other. So all the individuals in green shades versus yellow and red versus grays, and identify these subgroups and see why they're similar in addition to the fact that their epigenetic profiles might be similar. So we can then look at additional factors such as biochemical biomarkers, brain structure and function, cognitive profiles, and clinical profiles. So we started with a group of mixed individuals with various NDDs, and some of our expected results from using molecular profiling or epigenetic profiling are that we could use epigenetics to potentially differentiate between diagnostic categories. So we could see that the green group could be all the individuals that have ASD, and they have a different profile from the yellow and orange group, and those are the individuals that have ADHD, for example. But we could also see that if you can differentiate based on the epigenetic profiling, we could have individuals who are across disorders within a subgroup, but they may have similar features. So maybe everybody in the green group has a mix of ASD, ADHD, and OCD, but they all have sleep issues. So what are we actually using to molecularly or epigenetically profile? So going back to the video with all the different sort of chemical tags that are epigenetic marks or tags, our favorite epigenetic tag is something called DNA methylation. And so DNA methylation occurs at specific sites. It's very stable, and it's been extensively studied of all the different epigenetic tags. It's probably the most well studied. And it's found throughout the genome. It's not evenly distributed, but it is highly regulated. And again, a reminder that sort of in general, the concept of methylation, DNA methylation, is that it helps to determine whether or not a gene is turned on or off. So how do we actually measure DNA methylation in human tissues? So we can measure methylation in any human tissue. Within POND, we sort of focus on blood and saliva samples. Whoops, that came up. I guess they came up together, sorry. So we use something called a DNA methylation microarray chip, which is this figure at the top here. And it's this amazing technology where you can actually put an individual's DNA on one of those little squares, and we can measure DNA methylation throughout your entire genome just on that little square. And then we can quantify and then compare amongst individuals. So just to sort of give you an idea of what we kind of are looking for in terms of how we're measuring and quantifying DNA methylation. So for example, we have a gene X, and we have all those letters representing Oh, okay. Can I stop it from being on slideshow? Like pause it? No. Oh, okay. Okay. Okay, so we have, for example, a gene X, And on the left column, we have, for example, a sample from an individual with an NDD. And then on the right, we have a sample from an individual, a healthy control. And we can see that the methylation tags are distributed throughout that gene at similar places. So we would say that this is something they're similarly methylated. And then on gene Y, on the left, we see that the methylation tags are at different positions than they are on the right. And this we would call differently methylated. And so you would do this for pretty much all of the genes in our genome, and then we could compare genome-wide your methylation profiles. So some of the data that we actually have from the research that we've done within POND, first we looked at DNA methylation profiles of a mixed group of individuals with NDDs, so ASD, ADHD, and OCD. And then we compared their DNA methylation profiles in blood to those of healthy controls. And because they were mixed group, and I've already mentioned the fact that we know that there's so many mixed presentations and mixed causes, so the groups are very heterogeneous. They are similarly methylated to each other, and we can't use the DNA methylation profiles at this comparison to differentiate between individuals with an NDD and from healthy controls. However, when we look at subgroups within, for example, ASD individuals with a particular mutation in a gene called CHD8, or if we look at individuals with OCD and we look at potentially symptom severity based on the CY box score, which is an assessment tool for OCD, we see that when we compare the methylation profiles of those individuals in the subgroups to healthy controls, that they're differently methylated. And so using the epigenetic profiles here in these group comparisons, we are actually able to differentiate between those with an NDD and those who are healthy controls. And just to give you an idea of what the actual data look like, so our data don't look like the people and the little methylation tags, but so this is a heat map where each row represents a specific site throughout the genome where there is methylation, and the columns each represent an individual. And you can see that on this group here, the orange individuals here, who are individuals with ASD and a particular mutation, so the CHD8 mutation that I'd mentioned, they all have very similar profiles, so you can see that the pattern is very clean. Whereas the control individuals have a similar pattern to each other, but it's very different from those with ASD and CHD8. So we can use these to differentiate. In addition to using the DNA methylation profiles to differentiate between individuals with an NDD and without, they can also help us to understand the underlying biology. So we can look at the genes that are actually differently methylated to investigate the actual biological pathways that they are part of. And that will give us a better understanding of the biology that can lead to the development of an NDD. So for example, when we look in our data, we see that the differently methylated genes are involved in pathways that are involved in the immune system, in brain development and function, in regulating other epigenetic mechanisms, and in metabolic pathways as well. So we think that we can apply this approach to future studies within POND, where there's a wealth of clinical and other genetic information that we can use. And so, for example, we could apply this to looking at the DNA methylation profiles of all individuals, for example, with ASD and another specific genetic change, other clinical features such as sleep or GI issues, and then also looking at certain exposures to the fetus during pregnancy and also during early childhood. And finally, I just want to end with sort of broader questions that we're asking. So how can epigenetics help patients with NDDs? So some of the questions that we're asking and pursuing through POND research are whether or not we can use these epigenetic tags or DNA methylation as predictive biomarkers. So can we use them to diagnose earlier, and can we use them to aid in more accurate diagnosis as well? We also want to use them to complement and help to interpret unclear genetic results, because sometimes we have results where we can't interpret whether or not we think they'll be harmful. Can we use DNA methylation to predict responses to medication? And also identify new drugs and new pathways that we can target. And finally, can we actually correct these improper changes, epigenetic changes? So there are a lot of new technologies you may have heard about, CRISPR technology that can sort of correct genetic mutations, but can we correct these epigenetic mutations to improve outcomes? So you can see that from this list, we have a lot of questions to ask. I have a lot of work to do. We have a lot of work to do. I just want to acknowledge the research group that that have been working on these studies, and also to thank you for being part of our research. Yep. Oh, sorry.
I have two questions. First one is it a fair assumption to say that we can use the pond results to verify the DSM diagnosis? Because you just showed that we can separate the healthy group from the NDD group. And second question is there any evidence that ABA therapy can impact the epigenetic mutation?
Sorry, what?
ABA therapy or other
Oh,
interventions,
okay.
non-medical or
Right.
medicinal interventions can impact the
Right.
Epigenetic mutations.
Right. So for your first question, it's something that we would have to establish using our research first. However, I would say that the diagnostic criteria itself, as we had found, if we look at the diagnostic criteria, individuals with various presentations within, say, ASD, they're still so different from each other that we can't differentiate between all individuals with that diagnostic criteria and healthy controls. But there are subgroups within there where, for example, with the more severe OCD individuals with more severe symptoms, we see a better separation between individuals with OCD with severe symptoms and healthy controls. So we may be able to use it in addition to the other tools that are currently being used to sort of validate those findings, but we would need to be able to replicate this data in larger groups of individuals as well. But there is potential for that. And the second question about using non-medical and sort of correlating that with DNA methylation, that could very well be correlated. It's something that we could include in our research as well as a research question. That would sort of hinge on whether or not the ABA therapies are targeting pathways that are biologically relevant to epigenetics. So there are some that are maybe more relevant than others that we could look at. Yep. Oh, sorry. He was asking from before. Yeah.
Sorry. I was wondering as well, like, most of the research is based on physical things like diet and medication. Is there something maybe that could, let's say your mental state or your mental environment, let's say if you're stressful in the workplace, can that affect your genetic changes and things like that?
So your epigenetic changes, perhaps? Yeah. So definitely there has been some research in a mouse and rat model looking at maternal care, where the offspring, if they've received or not received sort of typical maternal care during the neonatal period, their epigenetic marks are different long term. So there's definitely a dynamic aspect of epigenetic changes as well as a more permanent aspect, and sort of the environmental cues that you're mentioning can also be studied in terms of epigenetics as well. So within POND, that sort of depends on what kinds of information we have that we can use to properly correlate that, but as a general question, yeah, it can definitely be. Yep.
Has much been done about finding the sources of these epigenetic markers, what triggers them to appear or not appear, and whether that can be modified?
So for sure, there's a lot of research. So these epigenetic marks are part of normal neurodevelopment or development in general. So we know a lot about the regulation for normal development. And whether or not they can be altered, there are, for example, known medications that change DNA methylation patterns. Whether or not they change them, so they're not being used necessarily to target those changes, when they may be a side effect of whatever else the medication is doing. But we can use, for example, if we find that there are a particular group of individuals with ASD and immune-related dysfunction, and that they exhibit a DNA methylation profile that's distinct, we can look at specific targets. So where are these differently methylated sites being are occurring? We can look at those genes and see if we can actually use any drugs or develop new drugs to target those pathways to see if we can actually change the methylation effects, to see if there are then downstream outcome effects, behavioral or physiological.